So I will start by just introducing the standard of care as it is now in the University Hospital of Leuven, in which I'm working. So basically for the small variants, so single nucleotide variants, and for the indels, we still use exome sequencing. We use microarrays or shallow whole genome sequencing for the detection of copy variants. And for some cases, depending on the phenotype of a patient, we will also look for short tandem repeats expansions using triple prime PCR. So you can see this is a wide variety of techniques. Some patients may have different tests, and we wanted to explore the use of long-read sequencing because we know that they can cover all those tests, basically. To start with, obviously we implemented our pipeline and our wet lab. So the first thing that we wanted to do was to check how our pipeline and wet lab was performing, and we used the HG002 cell line to do that. And we started with the structural variants because this is we know this is where we miss most of the variants with short read sequencing. And I represent it here on. Yes, you can see. I represented here the ideogram of chromosome 2 as well as the UCSC segmental duplication track. And then all the colors and here represents the density of the structural variant calls that we have detected using either mapping technologies and then one tool such as QTSV or Sniffles2 or genome assembly and again structural variant calling. What is pretty obvious is whatever tool you use and whatever starting point you have, whether you start from mapping or from de novo assembly, you do have always a very high density of structural variants at the repetitive regions, mainly telomeres and centromeres. However, if you look at the gold standard that is provided by Genome in a Bottle, first of all, the number of structural variants that you have would be much smaller, but also they don't cover the whole genome. Mainly those regions that are enriched with structural variant calls, they are not part of a tier 1 region, and these are all the variants that we cannot assess when we do the comparison with Genome in a Bottle. I also plotted here for your information the length, the distribution of structural variants, so insertion and deletions by length, just to pinpoint the fact that most of the structural variants that are included are mainly short structural variants. And also, you only have insertion and deletions, meaning that the variants such as translocation and inversions cannot be assessed with the current gold standard. That said, we still have those number of variants that we can test, and this is what we did. I will have 3 slides that are very similar now. Every time we check something, we plot the recall on the y-axis against the precision on the x-axis. You have to pay attention to the scale that is not ranging from 0 to 1, but I always zoom in the results. Basically, we want to be here at this corner, meaning that we don't have any false positive and we don't have any false negatives. And in this plot, I represented 3 different technologies of ONT because we are doing this exercise every time the technology evolves. So everything from ONT is in blue with different shades of blues. Then we have a PacBio results in pink, and the shape of a point represents basically the color. And I really like this comparison because you can see that the points are not clustering by technology but by tool. So actually the tool that you use for structural variant calling is more important than the technology that you will use. And both perform very well on those Genome in a Bottle samples. If we look at the single nucleotide variant calling, again the same graph with this time 2 different variant color, then you can see that you start clustering by technology instead of clustering by tool. But still, if you look at the scale here, we start at 0.995, meaning that we have already a very, very high precision and very high recall as well. I cannot say the same for the indels, but this is something that is already widely known for the ONT technology. But I also have to mention 2 things. The first one is that this is genome-wide, and we know that if we zoom in the coding variants, then indel calling is performing much better with ONT. And the second thing is that I will show later, most of the variants that we were looking at, we could still recall them. So it's not as bad as it looks. That said, we also wanted to look for the possibility of ONT to detect short-term repeat expansions, and we did that using real samples. This time, not more genome in the bottle one. This is the list of all the repeat expansions that are tested in diagnostics at the moment. And we selected 6 different diseases, 7 patients, mainly, so these are highlighted in blue, and we mainly detected those for which the STR was not exonic and where the expansion was really large. And to assess the results, I just plotted here on the x-axis the repeat length that we observe on the routine test, and on the y-axis the length that we observed for ONT, and you can see that for the dark purple points we have a very, very good correlation, and I will spend a bit more time on those points here. They are all at 100. This is the length that I arbitrarily gave to the sample because with a standard of care we knew there was an expansion, but we could not know what was the exact repeat length. So to me, these are also positive variants. And then we have this lighter pink 2 points. These are 2 alleles from one sample that have been genotyped 3 times with optical genome mapping, and every time with a different result. So for this, we know that there are repeat expansion. We know ONT is performing and detecting repeat expansion in the same range as optical genome mapping, but we do not know what the ground truth is. So these are, for us, positive results where we can see we can detect the expansions, but not only we can detect the expansion, but we also have access to the repeat content. So if we do the standard of care, we will have a result like this with a triple prime PCR. If we do the ONT sequencing, then we will also know for both alleles what is the content of the allele. I took the Friedreich ataxia example for 2 reasons. The first one is that you have first, A stretch, it's colored in blue, but I'm afraid the color is not that easy to see. And then you have a GAA stretch colored in purple. And you can see whether the expansion is happening in one or the other allele. And the second reason why I decided to show you this as an example is that it has been published that there is a hypermethylation of expanded allele, and that you can also observe easily on IGV here. So you have, for those who are not familiar, we have here an IGV screen of 2 alleles grouped by phasing, where all the first block here represents all the reads that are supporting the expansion, and you can see the different methylation parts, and the second part represents the non-expanded allele. Next thing that we wanted to check was the Episignature, and we use, in this case, real samples, cases, and controls. So it has been published already a while ago that some diseases have a specific Episignature, and 34 Episignatures have been published in this paper. And in this paper, they also said not only which positions were specific for each disease, but also what was the expected methylation value that we would have. So we wanted to check whether those Episignature could be detected using Oxford Nanopore. I give you here one example, but we actually did the same exercise for all the diseases listed here. So we extracted the methylation value at the loci that they published, and then we checked whether we could cluster our samples based on those values. And you can see that the Cordelia de Lange nanopore samples are clustering very well with the Illumina array samples here. We also did the same with a blind sample. Again, I'm sorry for the color, it's difficult to see, with yellow points, and it's also clustering with those. So this works very well. However, graphical interpretation is not that nice, so we also decided to automate the disease classification by implementing a support vector machine classifier. We used 40 controls and 18 cases, and we also used 2 cases for which we had variants of unknown significance. And this covered a range of 13 diseases. After our classification, the controls were properly classified as controls. We had no false positive, and 17 out of our 20 cases could be properly diagnosed. Looking more into the results, I plot here on the x-axis all the diseases that have been included, and on the y-axis the number of cases, and you can see that the colors corresponds to the classification. So in blue we have true positives, false negatives are represented in orange, and I also represent in green the variants of unknown significance. The first one I would like to spend slightly bit more time on is this one. This is a patient for which we had one variant in the ATRX gene, but we also had one mosaic variant in the KMT2. with the genes, I'm sorry, KMT2D gene, and this one is causing the Kabuki syndrome. And when we ran the classifier, this sample has been classified as having a Kabuki syndrome. So it confirmed that the variant for which we were not sure of the impact was really pathogenic, and that the variant in ATRX that was of unknown significance was not pathogenic. When we have these two mental retardation X samples, that were also run on the classifier and classified as controlled. We did the same exercise with the EPIC array, and with the array also no methylation signature could be detected. And there we have two different hypotheses explaining the results. The first one would be that the reference set that has been used to make the Episignature was consists of too few samples to be robust with ONT data. And the second hypothesis will be that the Episignature could be variant-specific, as we had here two missense variants. And the last one that I wanted to highlight is a female with variant on chromosome X that has been shown to be pathogenic, but the Episignature could not be detected by ONT, nor could it be detected by EPIC arrays. And then we here hypothesize that maybe the blood, that the DNA we took from the blood, the X carrying the mutation will be inactivated. And this could be checked by looking at the skewed X inactivation of the following genes, and it indeed confirmed that this could explain why the Episignature could not be picked up. So our results again were quite good for this part of the project, and we wanted to see, now that we have Episignature, we also have the genome data, so we can have a look at all the variants that we have, and we wanted to check whether all the pathogenic variants could be detected, and indeed all of them could be found. I show you here the distribution of variants, and this is what I mentioned before. We did have some insertion and deletions that were found to be pathogenic by other techniques, and that were found back in our cohort. And I also plotted here the length and the number of variants that were detected. So now I can go back to the last part of the presentation. We also have a small project on an unsolved cohort, again with real samples. Compared to what you have heard here, I think our cohort is pretty small, but still it's a very nice exercise and very nice cohort. So we have 27 trios suffering from developmental disorders. They have been studied using the routine diagnosis, but so far nothing could be found. And the hypothesis when starting this project was that the structural variant calling would be better with long-read sequencing data. The phasing will be better, and as I just mentioned, we also have Episignature that can be looked at. So we started by looking at the number of structural variants. Again, we had about 23,000 structural variants per individual, which is quite a lot, and filtering this is quite a challenge. However, we still have some nice positive cases. I show you here one de novo 11 kb deletion in one patient. The deletion is happening upstream the FBXO11 gene that has been associated with intellectual disability. This could be, it could match the phenotype, so it could explain the disease. However, there are still some functional studies going on to make sure that the expression of a gene is impacted in the patient with a deletion. Another example is a de novo inversion, where you can see here the two breakpoints. So you have one allele that is normal at the top and the one with the breakpoint at the bottom. And I show you here the two breakpoints. This inversion is disrupting the DYRKN1 gene. Again, it could explain the phenotype of the patient. And then we also have 9 base pair deletion. So this sample or this patient also underwent an exome sequencing prior to the long-read sequencing. And when we found these variants in this gene that could also explain the phenotype, we went back to the exome data, and we found that the variant was indeed detected, but it did not pass our QC, so we could not be, it was not interpreted at the time. And now with long-read sequencing, it passes the QC, and we can have a look at it. And the last example I wanted to show you is the case of two inherited variants, actually. You have one single nucleotide variant that is inherited from the father and one 4 base pair deletion inherited from the mother, that again is happening in a gene that could explain the phenotype of a patient. So taking all those results together, plus one sample that I did not mention, but for which we found Episignature in our cohort, but we are still looking for the causative variant, it will mean that we have a diagnostic yield of 18.5% in our small cohort. And you have to keep in mind that 22 patients are still being analyzed, so this could still increase or decrease if we cannot confirm everything the way we want. To finish, I wanted to have just a few slides on complex structural variants. So I just showed you everything is working very well in our hands, and we are happy with the results, but not everything is as easy as it seems. So we have this male patient, and we look at the data from our pipeline, and we detect 115 kb inversion on chromosome X. We do not think that the inversion could explain the phenotype because this has been inherited from the mother and the mother inherited from her grandfather. But still, if you look at the data, it does not seem to be an inversion. So if we translate the call, it will mean that we have before the inversion a normal sequence, then the part that is inverted in purple, and then the part in green that is normal, and then we should have a normal coverage. To make sure what was going on, we did perform optical genome mapping, and the optical genome mapping showed that actually the part in green was duplicated, which was not what we expected, and by doing manual inspection over the reads, then the hypothesis was that actually the purple segment will be split in two, and only one part of the purple segment will be inverted. We do have reads supporting most of the breakpoints, but not all of them, so it's not fully resolved yet. And we also try to perform de novo assembly on this sample to try to resolve it, but unfortunately, the de novo assembly did not give any nice results in this region. To conclude, I think I could show you that we have a very good concordance between our standard of care and the ONT data that we have, both for STR expansions, but also for the detection of episignature. And we also could find all the pathogenic variants that we previously identified. ONT also offers the additional benefit that you can classify variants of unknown significance in case of episignature, and you can look at the X inactivation. And finally, with our small cohort, we did have an increased diagnostic yield over standard of care. And I would like to thank all the people who have been involved in the wet lab interpretation, patients, clinicians, everybody. Thank you.